Falaschi's scientific biography coincides with the history of molecular biology and genetic engineering, and especially with the efforts undertaken here in Italy to establish excellence in this sector. So he graduated in medicine in 1955 and began his activity as a researcher with the biochemist Enzo Boeri in Ferrara, with the geneticist Adriano Buzzati Traverso in Pavia. He embraced what was, at the time, the new world of molecular genetics in the United States during the years 61-65. He worked at the University of Wisconsin with Hargobiin Khorana, a Nobel laureate in 1968 for deciphering the genetic code. In Stanford with Arthur Kornberg, 1959, Nobel laureate for his studies on DNA replication. Upon his return to Italy in 1965, he was one of the undisputed pioneers of molecular biology in Italy. Professor of molecular biology at the University of Pavia for 12 years. In 1970, he became director of the Institute of Biochemical and Evolutionary Genetics of the National Research Council, the CNR, in Pavia, founding the current headquarters and substantially promoting its international scientific growth until 1987. During the years in Pavia, he was successfully president of the Italian Society for Biophysics and Molecular Biology, president of the Commission of the Italian Ministry of Health Study on Recombinant DNA, and became a member of the European Molecular Biology Organization, EMBO. From 1982 to 1989, he was director of the CNR Finalized Project on Genetic Engineering and Molecular Basis of Hereditary Diseases, and from 1985, vice president of the National Committee of Biotechnology of the Ministry of Scientific Research and Technology. From 1987, he embraced the cause of the International Center for Genetic Engineering and Biotechnology, which was a young institution with a lot of money, but no seat, no laboratories, no people. And he assumed a fundamental role in establishing the headquarters of the center in Trieste, creating first the laboratory in Trieste and then the laboratories in Delhi, and promoting the center overall development and international reputation. Starting from scratch in 1987 under his guidance, first as the director of Italian component and then director general. Between 1985 and 2004, ICGB grew progressively to what I just described you today. During the years spent in Trieste as Director General of ICGB, his cultural commitment for the promotion of scientific research in Italy remained unflinching. He served as a member of the National Committee for Biosafety and Biotechnology of the Council of Ministers, member of CNR Board, member of the National Council for the Investment Fund for Basic Research, the FIRB project here in Italy. In 1998, he received the gold medal and the first-class diploma for cultural and scientific merit from the President of the Republic of Italy. In the international arena, he was a member of the Third World Academy of Science, a member of the European Science and Technology Assessment of the European Union, and he showed a great commitment to the Human Frontier Science Program Organization, and he was chairman of the Council of Scientists from 1999 to 2001. Upon completion of his mandate as ICGB Director General, Falaschi remained associated to the center as a distinguished scientist, moved his research activity to the Scuola Normale Superiore in Pisa. For those of you who are outside Italy, Scuola Normale Superiore is one of the leading universities in Italy. He was professor of molecular biology, director of the Laboratory of Molecular Biology of the Scuola Normale. At the same time, he continued his international activity. He was executive director of the Asia Pacific International Molecular Biology Network, with headquarters in Seoul, in Korea, and rector of the International Center for Chemistry and High Technology, the ICS UNIDO, based in Trieste. There have been a number of obituaries and articles written to celebrate Arturo's accomplishment and vision. I would like to show you just one, which is this one, which was published last year in the Corriere della Sera, which is the newspaper with the widest readership in Italy. It's signed by Claudio Magris, a famous historian and writer here in Italy, which underlines one of the most striking capacities of Arturo, namely his way to couple advanced science with a genuine and deep interest in humanities. Very humbly, however, the way I would like to remember Arturo is by trying to summarize what he would have considered, at least to me, who have been, as you have heard, a young PhD student with him, his take-home lessons, namely some of the qualities that distinguished him and probably which were the one he wanted to pass to the younger generation. And the first take-home lesson is to put passion in everything you do. The passion for Arturo was certainly science. He was always present in the laboratory, paying attention to very small experimental details, being able to enjoy small advancement in each of the research projects his younger PhD students and postdocs were carrying on. There were always copies of Nature, Cell, Molecular Cell, and Science on his desk, along really a myriad of books on science. He was purchasing on Amazon and other providers. I really don't know where he found the time to read all these books. A second take-home lesson was be tenacious. None of what he accomplished would have ever been possible without a very strong will and determination. He accepted the director of ICGB in Trieste when this was little more than an acronym and an idea. I remember visiting him in Trieste the first time. There wasn't just an empty room, a desk, and the phone on the floor, because there was not even a place where to put the phone, and the line was not sufficiently long to put it on the desk. He threw really basically himself into this undertaking, dedicating body and soul. He built the center step by step with vision, foresight, and an unstoppable tenacity and an extraordinary enthusiasm. A third take-home lesson is to cultivate good friends. I had the privilege of meeting Arturo more than 25 years ago, you have guessed, when I was a young PhD student in Pavia, and I remember even now how the first time I met him with a lot of sheets of research data, the BRDU gradients at that time. He was a scientist in the field of DNA replication. I was really fascinated by the charisma of the scientist, and at the same time by the simplicity of the man. And the enthusiasm he transmitted was really overwhelming. He had an extraordinary charm, a very humble manner, and was endearing really to everyone. Here you can see him with a series of Nobel laureates. However, you can trust me, at the same time he was able to establish the same type of very friendly relationship with really everyone, and including many of the colleagues that I'm seeing here, and young people I'm seeing here in the audience now. A fourth take-home lesson from Arturo is to pursue excellence. His scientific contribution to the field of DNA replication has been numerous and important, and this has been a research theme in which he put really all his patience in the 60s when he returned back from the United States. He developed procedure to isolate newly synthesized DNA by gradients, as I was saying before, and thanks to the method that he developed, he has been the first one to describe the first human origin of replication of the human DNA, the lamin B2 origin that now still remains the model origin in the DNA replication field. Consider that since 1994 up to now there have been only 30 origins, or more, roughly 30 origins described out of 30,000 origins, to let you understand how difficult this field is. And another interesting observation is that he remained really one of the very few international researchers whose scientific activity is testified by publication in PubMed through almost 50 years, so from 1962 to 2010, when he passed away. And the last message from Arturo is always embark in big projects, so have a vision in everything you do. Without this particular vision for ICGB, we would not really have not been here to celebrate this moment today. We wouldn't have had, as Professor Barari reminded, 740 fellowships awarded over the years, almost 20,000 participants in the meeting and courses, more than 2,500 papers in Medline published by and signed by ICGB scientists, and I have to say that his vision drives much of the work that we continue to carry out today. The reason we wanted to find something special for him, also scientifically, so to dedicate to him this series of meetings on molecular medicine, is based on the fact that he always had a vision for recombinant DNA becoming something important for molecular medicine, which is obvious now for us, but it was not obvious in the past. This is, for example, one of his publications in Italian from 1979. 1979 was only 2 years after Sanger, Maxam, and Gilbert designed the first techniques to sequence DNA. It was only 4 years after the first southern blotting was performed. And already at that time, where it was not clear how much DNA there was, how genes were built, and how to continue research in this area, already there he thought that all this research on DNA could go forward molecular medicine. So it is right in this spirit that we believe that the idea of having this series of meetings on different topics every year on molecular medicine is something that honors his memory and his vision at that time.